Okay, the basic unit of a muscle, the sarcomere, the muscle cell itself. And by definition, it's from Z line to Z line. These are structures we see when we look at the cell under the microscope and such. So you can't see it under a normal microscope. You have to use an electron microscope, because if you look there, 595 nanometers, that is pretty small. Such. But if you look right here, here's a line right there, then there's a line. That is known as the Z line. Okay, that's the end, the Z line. And then attached to the Z line is the actin, the thin filament. Okay, now down the middle, where'd my cursor go? Down the middle of the cell is the M line. There we go. And right there, that is the anchoring point for the myosin. So the myosin is anchored to that, and that creates the A band. You can see it in the brackets right there. That's the darker band. It's darker because it's thicker, so it absorbs more light, or it prevents the light from passing through, so it appears darker on the microscope. Okay, so the I band is the lighter or the paler zone, and then the A band is the dark center region of the sarcomere right there. Then the very middle, which is a protein, is the M line right there, and that's what the myosin anchors into. And then the myosin pulls. It pulls in from both directions simultaneously, and it's trying to pull the actin over itself, causing the Z lines to move toward the center, toward the middle, such. In the very center is the H zone. Okay, now if you notice on the I band, it has a dark, then the M, and then the dark. That middle area right there, that's the H zone. And that is just myosin. On the edges where it get the darker banding, that is where, because the myosin and the actin are overlapping and causing even a thicker area, which prevents light from passing through, so it appears darker. So the way this whole thing works, that when a muscle contracts, the filaments slide over each other. The actin slides over the myosin. The myosin's pulling it, but it slides over it. So when it does this, when the sarcomere shortens, it's because the myosin is pulling the actin toward the middle. And as it contracts, then the H zone goes away. It becomes darker. It all becomes the same consistent color, if you will. The I band on the side that's being contracted, it goes away. The whole I band might not go away, but the part that is actually sliding in, it'll go away as well. So that varies in its color or appearance. So the proteins involved in doing this is the thick filament myosin. That's the A band. It is a helical protein that has this globular head on the end of it, and on that head is an enzyme known as ATPase, and that ATPase is what's going to allow it to take in ATP and break it down to ADP and phosphate, which then allows it to change its shape or to change its conformation. And it has an affinity. The myosin has this affinity to want to stick to the actin. Okay, there's an actin area that's built for it to attach into. So the thin filament is the actin. When you see that G right there, that means natural or normal and such. So it is a globular protein, and each little glob has a little site on it that myosin wants to stick to. That's known as a myosin binding site. Okay. There is another one, tropomyosin, and this wraps around the actin and it covers the myosin binding sites. Then attached to the tropomyosin is another protein called troponin. and it's the troponin that the calcium binds to. And once the calcium binds to the troponin, this whole thing starts and such. So the thick filament is myosin, and the thin filament is these three other proteins, the actin, the tropomyosin, and the troponin. Okay, so the troponin is a series of proteins, and when the calcium binds to it, it sticks, but the calcium can leave. So it's a reversible attraction, if you will. So when the calcium binds to it, it changes its shape. The fancy word for shape is conformation. So it changes its conformation. And when the troponin changes its shape, it causes a tropomyosin to move. And when the tropomyosin moves, it exposes the binding sites on the actin. When they get exposed, the myosin sticks. And that's the beginning of the twitch. Okay? So the myofibril contains the sarcoplasmic reticulum, which is the cell's ER. And it also contains these T-tubules. The T-tubules do not carry anything. They're little hollow tubes that pass down through the muscle. They conduct the impulse, the action potential. So you get an action potential to show up. It travels across the surface of the sarcolemma, which is the membrane, and it travels down through the muscle cell through the T-tubules, causing the SR to depolarize. When it depolarizes, it releases calcium. The calcium goes from high concentration in the SR to low concentration in the cytoplasm, and it travels out and it sticks to troponin. If you remember the type 1 and type 2, the type 1 didn't have much of an affinity for the calcium, its troponin didn't. And the type 2, the troponin had a very high affinity for the calcium. So the calcium travels out and it leaks out into that area and it attaches to the troponin. The troponin changes its shape, which causes tropomyosin to move, which then opens up the binding sites on the actin. The myosin sticks. It goes in there and sticks, and as soon as it does, remember it has that ATPase on it, it sticks, and that causes the ATP to change to ADP and phosphate, ADP and PI. So that causes, when that changes like that, it causes the myosin to change its conformation or its shape, and it swivels. So the myosin binds, and then the myosin swivels. That 5, 6, and 7 all happen simultaneously, if you will. Okay, the swiveling occurs because the ATP changed. It went from ATP to ADP and phosphate. So the myosin swivels. Then that ADP and PI are released. They go away. Then, and only if ATP attaches, will the myosin release from the actin, change its shape to like it was at the beginning, and then reach up and bind again if the binding site is open. And if it binds, then it, boom, ADP and PI is formed, and then it swivels. And then the ADP and PI go away. And then ATP attaches again, and it just keeps cycling from 9 back to 5, 9 back to 5, over and over and over, until you run out of ATP or you stop the action potential. Then why did you stop the action potential? You say, hey, stop moving, or a signal came to your brain and said, ouch, stop doing that. So you had a pain signal come in and tell you to stop doing that. So that's the steps in the contraction cycle. And that's what you kind of need to work on and get those steps down. Okay? All right. So we're going to stop right there.